Hello, everyone, and thank you for joining us on this session. I'm very glad that here with me are Todor Primov and Martina Markova, who Todor Primov is the head of life science and healthcare at Ontotext, and Martina is a product manager at Ontotext again. So I'm very glad that they are here with me. Today, they are going to speak on the topic of revolutionizing the knowledge graph-based application development with LinkedLifeData inventory, which is going to be a very interesting topic and presentation. So without wasting additional time, I'm giving the floor to them, but before that, I will just let you know that if you have any questions, you can ask them by just typing them in the chat box in the panel on your right. So with that, Todor, Martina, the floor is yours.
Thank you, Yasen. I hope you can see my screen. Well,
Yeah.
the title of the presentation is quite long, but actually in a simple words, how we can benefit from our LinkedLifeData inventory in order to build quickly, faster, high quality knowledge graphs that can be used for various use cases and can supply various systems with structured knowledge that you can use more efficiently. This is quickly the agenda for the presentation today. Why knowledge graphs? I assume there is no need actually to strengthen this message to the audience because you are all convinced about the power of knowledge graphs and how we can efficiently use them in multiple various domains, like in our case, life sciences and healthcare, where we can solve different challenges. Then we'll go to some specific use cases that we have implemented on top of LinkedLifeData inventory, and with a special focus on one of our products named Target Discovery, which will be covered by my colleague Martina later on. And then we have a quick Q&A session at the end. Why knowledge graphs? For me as a biologist, actually, this is the normal way how we can store and represent knowledge and information. Luckily for our particular domain, we have a lot of reference data sets, ontologies, terminologies that we can use in order to disambiguate each of those concepts, objects from the knowledge graph that we can use in different contexts, which is defined by the connections between the different concepts. And really to put all this information in specific use, in specific context, based on the surroundings of the concepts in the knowledge graph. And these knowledge graphs can be used in many different ways. They can be queried as a standard database. You can construct very complex queries that can traverse the network of knowledge in order to identify specific subsets of objects in order to satisfy your data needs. Or you can do some more complex, very complicated analytics on top of the knowledge graph, including also knowledge graph embeddings. You can predict new type of relationships between the nodes, or you can even predict not existing nodes in the knowledge graph. And for that purpose, we can use also heavy logical instructions provided by the ontologies that can help us to infer new knowledge, new facts. Well, in the life sciences, we face various challenges. I will focus on specific ones which are determined by the nature of the data that we are using for our research and investigations. So we are proud that we have really boom of data. Every few years, the information, the amount of information available publicly doubles, which is absolutely great, but also introduce new type of problems. We need to have new technologies, new tools that will help us efficiently to query, to analyze, and to use this data. And one of those tools sits on top of knowledge graphs. And the technology behind it, which allows us to unlock the information hidden in many different diverse data sets, to build a network of knowledge, to provide the data in a way that it's findable, accessible, interoperable, and reusable, the FAIR data principles, in order to solve various recurring issues that we find in every single domain in the industry, in every single use case, that we need to integrate heterogeneous, disconnected data sets, convert the data in a unified format that will help us to more efficiently integrate it, to connect it better in order to be able to reach to a much deeper insights, and to automate the whole process and to ensure quality on every single step within the process. We've solved various type of problems, then can be split into two major groups: technicals and qualitative problems. Uh, the technical cover mostly the obstacles on a daily basis that we have, again, disconnected data, different formats, different update intervals, and the volume of the data. While qualitative, again, they cover discoverability of the data. In most of the cases, the data is not very well described, so it cannot be understood what type of information each of those data sets contain, or it's not very well normalized, so you cannot efficiently use it either for information discovery, but also for any downstream processing steps that can follow afterwards. Yeah, so knowledge graphs are actually quite good choice for traversing huge networks of knowledge that will allow you to extract deeper insights that can bring you to a competitive advantage in many use cases and scenarios, because, well, the information is very well integrated. All the different type of relationships between the main classes are identified, structured, materialized. You can traverse the graph. And on the other hand, in addition to the data itself, we provide also all the necessary metadata describing the data sets, so you are aware what each of those data sets contain, what was the origin of the data, who owns the information, how you can access it, so that you can reuse it in any other use cases later on. The technology itself, RDF and knowledge graphs, they demand high quality of data because we don't introduce unstructured content or higher volume of unstructured content in the knowledge graph itself. We always aim to normalize the data to reference ontologies, terminologies, and other data sets, so that this translation of strings to things will actually enable any other downstream processing, meaning machine learning, deep learning methods, to work on the knowledge graph and to infer new data and to actually use it more efficiently in the analysis. So the link-wide data inventory allows you first to be much more faster, to deliver solutions 3 times to 5 times faster compared to starting from scratch, identifying the data sets, translating them into a unified format, integrating all the data, and then start building the application. And it's also very powerful in terms of decreasing the cost of development of such applications, because all these components can be easily integrated directly off the shelf and you can start using them immediately. So actually, what is link-wide data inventory? This is collection of hundreds of public data sets and ontologies that we have accumulated through the years working on various projects. They are already in a semantic ready format, either in a custom RDF or some of the OW, OBO, other formats that those data sets are distributed. And we can cover with those data sets various knowledge domains in the life sciences and healthcare domains, ranging from genomics, proteomics, metabolomics, molecular interactions, pharmacology, clinical, medical, and scientific publications. You can think of various, the well-known data sets in these particular domains like UniProt, Campbell, KEBI, Unified Medical Language System, all the medical terminologies like SNOMED, CT, LOINC, ICD-9, 10, also some specific domain ontologies like gene ontology, cell ontology, other terminologies used efficiently quite often in the domain. We also include some more niche ones like genome-wide association studies, DisGenet, Open Targets, clinicaltrials.gov, Pathway Commons, and some data sets from FDA like drugs and FDA national drug codes. The product itself, it's a solid data integration platform that we use our proprietary Ontotext methodology for semantic data integration and building knowledge graphs. In the core of this methodology stays the verification steps that we have defined in the various workflows that ensure semantic harmonization of the data, normalization of all the properties, to reference terminologies and ontologies, and we augment the publicly available data where necessary, of course, with additional semantic mappings that will improve significantly the connectivity between the datasets in the inventory, will allow actually the traversal of the knowledge graphs that you built with in the inventory with those additional links that bridge the gaps between the datasets. Of course, there's a possibility to enrich the proprietary data that you have with the reference terminologies and ontologies from the LinkedLifeData inventory. The very important part of the inventory is the so-called semantic data catalog. As I mentioned already, as certain steps in the workflow ensure the fair data principles implementation on top of the data sets that we have. At certain steps, we generate the full description of the data set in terms of data catalog vocabulary and also full documentation of the classes and the properties for each of those data sets, referring to void schema, which will give you the full context, the full content of the data set. And one very important aspect is the automation. All the workflows that we define for each of the data sets in the inventory are fully automated from starting from the automatic download, transformation of the proprietary formats to RDF representation, then generation of the DCAT and void, and also the additional semantic mappings, and then publishing the data set as part of the data catalog. Very quickly, few use cases that were built on top of the LinkedLifeData inventory. The first is we have built together with a big pharma company a multimodal preclinical knowledge graph integrating data from 8 public data sets. You can see which are those. And we started with the initial semantic data integration steps, then alignment to the customer domain model, and improving the interconnectivity of the data sets with additional mappings that we already have in the data inventory as part of each of those data sets. So within less than 2 weeks, we managed to build a knowledge graph of close to 1.5 billion triples, facts, and significant amount of the data, around 40%, was actually new inferred data based on the ontology that we included in the knowledge graph. So in short, very quick development process. You can demonstrate for less than 2 weeks benefits of integrating highly heterogeneous data sets into homolytic knowledge graphs, and then starting the data exploration journey with building different dashboards and analytics on top of it. The second use case is developed together with one of the biggest US child hospitals, and actually for their research institute. It's a more or less a Doctor 360 view over the research scientists in their research institute. So usually the information about the research for each medical professional is scattered around different systems and data sets, starting with the internal data sets in which they have partial information of the publications and the work done by the researcher. Then we augment it with public data sets with publications from PubMed, Microsoft Academic Graph, later on replaced by OpenAlex dataset, Crossref, clinicaltrials.gov, in order to identify what were the outputs from the research work of these scientists, and also what were the collaboration patterns with other researchers, other research institutes, what are the research topics, and so forth, based on the interest of their management. Very quickly about the third case study, which is very similar to what Martina will present you in a minute. It's a project with a US-based biotech company for which we have developed a huge knowledge graph, which is used to identify new potential targets for new drugs. And in addition to the huge amount of data that we integrate from more than 35 different data sets, we also augment the knowledge graph with significant amount of data extracted from scientific publisher with NLP methods. So this actually significantly accelerates the research, provides really very large complex networks that needs to be analyzed to identify relationship between different biomedical entities, which allows data-driven decision making of the company researchers, and will allow at the end to identify those highly valuable potential targets, and to start to continue actually the next steps. And one of the significant, as you're going to see later on, advantages of the system, it's highly adaptable based on the specific needs of the end users. And now I'm going to step back and provide the floor to Martina to explain you more about the target discovery.
Thank you, Todor. Let's kick it off with the next slide. Okay, so what is the aim of target discovery? It's a well-known issue that currently the process of delivering drugs to the market is extremely complex. It is estimated that it costs more than 2.6 billion and almost 20 years to deliver a drug to the patients where they need it. Why is this? Because the whole pipeline that is required, it's so huge, and there's so many different steps and so much that can go wrong. And the statistics are actually pretty gray. Only 1 in 10,000 compounds is estimated to actually become an approved drug after it goes through all of these required steps. And oftentimes, early in the process, there are a lot of different issues that are encountered and that can be prevented, so that later on success is more probable. Number one is that actually researchers nowadays need to build better hypotheses that are based on sound biological data. And as Todor already mentioned, data grows by the minute. And it is crucial to find which of it is actually usable and which is of high quality and can be applied to specific use cases. The next one is how do they combine all of these parameters that can be encountered in the different data sets, and what analytics can they run so that they can come to an actual insight. Of course, early target termination and target prioritization is essential because usually among all of these data sets, people will encounter many different targets that may seem viable, and helping them to concentrate on the ones that are truly significant is essential so that they can put a focus in their efforts. Then oftentimes the confidence in their decision making is not high enough, primarily because the data quality is not sufficient or because they need so much effort to dive deeper into each potential target. And then, because we talk here about such a huge timeline, companies do need to be flexible throughout all of this time and be able to pivot and be able to change their own strategies based on new data, new findings, or what the market is doing right now. Next slide, please. So where do we integrate in this whole process? We are into the preclinical steps, and what we do is to cover the target discovery part, where we help researchers to discover genomic and proteomics information, to combine data from public databases like UniProt and NCBI, but also from textual data like PubMed, and to uncover targets which are really significant for a disease. Then we do a small part of the target validation, meaning that we prioritize the targets based on known facts. We also help with lead generation and refinement, so we can help uncover compounds that target these specific proteins, and also cell lines which are commercially available and can be purchased for further experimentation, and literally deliver a full report that can be then used to perform the wet lab experiments which are required to validate all of the findings into a single system. Next slide, please. So what approach do we take? Is you can already imagine we're using the LinkedLifeData inventory. Currently, it contains more than 200 data sets which are integration ready. Actually, our solution uses way less than that because it's not necessary to integrate all of this, but our customers have their full options, so they can choose what to integrate. Oftentimes, they choose about 30 of these data sets or even less, and they prefer to integrate also their own proprietary data. A lot of the companies and partners we work with actually generate their own internal genomics expression data, and they contextualize it by using the knowledge graph, which contains also fully mapped relationships. Apart from that, we integrate more than 80 million text documents, such as scientific publications, patents, and annotated clinical trials, all into the same graph. All of this data can be used then to make predictions. So the data that Toller already presented is not just integration ready, but it's also AI ready. So based on this, we can make predictions about gene-disease relationships or protein-protein interactions. And we can use various types of analytics to truly drill down this information to what matters most to our customers and their use case. And inside the target discovery application, users are mostly interested into information retrieval so that they can truly dig into the facts of each of the targets that they're interested in. They have powerful visualization tools so that they don't need to be hardcore data scientists. They can visually already analyze the data. There are also some deep data analytics for the data scientists out there who are familiar with some of the algorithms, or if they have developed their own, they can also integrate them into the tool. We do have automatic target ranking so that researchers are able to actually in bulk already find patterns among thousands of different targets with the click of a button. And we do provide confidence and provenance at every step because data quality is of utter importance to our users. Next slide, please. So the AI-powered gene disease predictions are one of the most powerful tools that we offer to our customers because it unlocks information that is otherwise not available. Most of it is, not most of it, but about 100 of each of the sets of predictions that we have made by using different methods are also manually curated. So we have checked whether this is actually true, what we have already predicted. The training data is regularly updated, and it's also configurable so that we are also pretty flexible and we adjust in time to the growing amount of data and new facts which are published. And we do have also graph embedding models. So what does it mean is that we take the information, we represent it as a vector, and we are able from this to do various other types of things like not just predict gene disease relationships, but also find similar targets to ones that are already successful within a company, or similar compounds to the ones that are already drugs that have made it to the market. And we also do text mining. I'm just going to repeat myself. We do NLP on top of about 80 million scientific publications. And this information can be integrated into the same knowledge graph, providing even richer context and even more inferred relationships. Next slide, please. So what are the benefits? A number of them. Actually, we have estimated that following the target discovery procedure and workflows, customers are able to save hundreds of millions of dollars per project. Why is that? Because if we de-risk early on the whole process, this would down the line of every step of this huge process of drug discovery, including the clinical steps, can save a lot of dollars that would have been otherwise bound to fail. Direct benefits that are also accounted for is the shortened time to market. Actually, reports and recent publications show that we do have usually 3 to 5 years that are required for preclinical discovery alone. And one of our customers recently reported that they just need 18 months in order to go to the clinic with targets. Then drug repurposing unlocks new doors and new sources of revenue for huge pharmaceutical companies who already have drugs on the market. Also, data scientists and data analysts can be more efficient, and they can work better together with the actual subject matter experts, such as scientists and researchers. And of course, all of this boils down to optimizations, cost cuts, and saving times of the actual scientists. Next slide, please. Who is it for? It's primarily about researchers and translational medicine experts. So the application doesn't require any technical skills to get going. They're pre-configured analytics and visual dashboards so that any scientist can work with the data without requiring any coding or any Spark or SQL knowledge. All of these configurations can be also further enhanced to fit with each individual's use case. And this also helps the researchers to be more innovative and to really quickly build novel hypotheses just by looking at the data. And of course, for larger corporations who already have established data scientist team, this tool can help them to better collaborate and to do configurations on their own, or to integrate their own algorithms if they have already developed such in-house. Of course, the data quality is also higher because we're working with data inventory. So a lot of the steps that data scientists might usually do, for example, during an ETL process, are already spared, they're already done. Next slide, please. So we're almost at the end of the presentation, and here we have two case studies. Number one is with, again, a US biotech company. It's focused on immuno-oncology, and it already got a significant investment. It has already a well-established pipeline of targets, and they already got into clinical trials with one of them. And before using the technology, they used to do a lot of manual work. And for each of the targets that they had in their pipeline, they had to spend several hours and several days to retrieve all the information necessary. They didn't use any central database, so they were just going to each of these databases like UniProt and PubMed and clinicaltrials.gov separately. They had to manually gather all of these data and to align it and process it and analyze it and share internally with the team. And the whole decision-making process was taking weeks. Then they started using the solution with the knowledge graph power target discovery. So at first there was no NLP integration, but it was still really useful to them and they saw a lot of value. So what we did is to choose some of the data sets and to advise on the most important ones for their use case. And these were about 35 data sets. And we also did a lot of mappings between them. And these mappings are actually expert curated. So subject matter experts were doing them, which also guarantees higher data quality. All in all, the knowledge graph grew to be about 5,000 million facts big. So these are like billions of facts integrated all in one place. And alone, this already helps the researchers to be way more efficient because they can find all information that they need in one place. There are also additional insights that are inferred. So the innovation potential also grew significantly. And then they decided also to add NLP-generated data from about 40 million documents, which generated further 500 million facts. All of this was also integrated into the same knowledge graph. And all in all, they managed to find all the information that they need in just a couple of minutes per target. And even though we're talking here about target, actually the power of target discovery comes from the fact that they can do analysis in bulk. So they have so far analyzed hundreds of targets all in the same time in a completely automatic manner. So they don't do the analysis one by one, but they do it in bulk. Next slide, please. And here is our newest partner case study from Queen's University in Canada, the Mulligan Lab. It's again a lab that works in oncology, and they're particularly interested at protein called RET. It's well known to be vital for some of the oncological processes. So it's a well-known protein in the context of cancer, and there are already therapies that target RET. The problem is that many of the patients actually don't respond to these therapies, which is why scientists are looking around this protein to find other interactants that might be beneficial to target, because that might help these patients that are non-responders. The challenge for the lab that we're working with was that it managed to generate a lot of internal data. So they now have more than 2,000 genes. And this is just huge amount of data to be analyzed. It's difficult to screen all of these genes one by one and to prioritize them and to really see what is their role in the red pathway and in cancer. So what we did with the customer together is to first have one call in which we learned everything that I just told you. What do they do? What is their challenge? And we define how can they help in their particular use case. Then we integrated the proprietary data from gene expression. Then we set up the ranking methodology, and the lab tested it on top of 85 of these 2,000 genes or proteins. And they were actually really happy with the first iteration. They still did two more so that their ranking can be further enhanced. And after that, we're going to scale all of these to 2,000 plus candidates. Why are they happy? Because within just 3 days of usage of the system, they already found a therapeutic candidate that was not uncovered before. So they found actually a pretty valuable insight, and they did it really fast. So the customer reported that usually such work takes 2 to 3 weeks for just a single target, but this time they managed to do it just in 3 days. And like I said, it's really scalable. So they did it for 85 proteins, and now we're going to scale to all 2,000 with the same methodology. So within one click, they'll have their result. And that's all there is to it. We're now open to any questions if there's time.
Martin, unfortunately, we are right on time. Actually, we are overpassed the time. So all of the questions will be follow up. I think Toshko already did follow up on the most of them, I think. So thank you both for the very interesting presentation. And let's jump into the next one. Thank you.
Thank you. Bye-bye.
Bye bye.